Okay, so here's the next thing. So protein structure, and we're going to be running into a lot of proteins and talking about their structures. It's kind of unavoidable. This is the level at which you have to work to understand vaccines, but this is another part of our just little getting everybody up to steam. So there are four different levels of structure within a protein. And they're just different ways to think of the protein, really. So level one, you've got your primary structure, which is just the order of the amino acids. So a protein is usually made of just one chain of amino acids. And it's just like a chain, just like the chain you're thinking of. The amino acids are all in a row, and then those rows can coil up into an alpha helix, or they can bend around and be really straight and be called a beta strand. But the actual order of the 20 different amino acids, however many in whatever order, that is the primary structure. So that's not even really structure. That's just like, the order of things in the protein. Order of amino acids. All right. So, and we can figure out what a primary structure is going to be just by reading the DNA or RNA, because that tells you exactly what protein has to be made from this. RNA and DNA are instructions to make protein. Next level, secondary structure. All right, we already talked about this. Secondary structure is it alpha helix? Is it beta sheet? Beta strands make up a beta sheet. Or is it random coil or, flexible loop? But let's just call that random coil. We'll lump it all in as that. And these three are the three secondary structures. So you can imagine your long chain of a protein, and now it's going to have some bits that are coiled up and some bits that are folded back and forth to make a little beta sheet. And it's still that same long string. Like you could do this with a piece of spaghetti if it was very nicely cooked. Maybe a little better if it's overcooked, actually. Yeah, and you can make yourself a nice little protein. You can do it with a piece of string, you can do it with a cord, whatever you like. Third level, tertiary structure. Yeah, okay. So another word for this is the fold. And this is just the three-dimensional arrangement of the alpha helices, beta sheets, and random coils that are made up of the amino acids in the primary structure. So the tertiary structure is really like the three-dimensional shape, the 3D structure. And so you'll have particular organizations of these things. You would think that anything might be possible, but the more you study proteins, you realize there are actually only a few of these shapes out there that are actually useful, or at least are actually used in biology as we know it. And so, yeah, when we're talking about a protein fold or a protein's overall shape, this is what we're talking about. And then the fourth level, the quaternary structure, which just means fourth level, is that the way that those three-dimensional proteins stick together, because most proteins don't work very well on their own. Usually they have to club together and build some kind of a machine. I don't know if you're fans of ancient anime, but it's kind of like Voltron, where the 5 little components would come together and make a cool space robot. That is exactly how proteins work, and yeah, it's how just great anime works as well. Or whatever that was. I guess that was maybe more American. All right. Now, so these are the concepts. Now let's look at the concepts in pictures, because that makes everything better, I think. I'm a very visually oriented kind of learner. Okay, so we'll do an example, sort of general example, then we'll do a super duper specific example where we go into ultimate detail. So, all right, I think you're going to like this. All right, so step one. 1 with a little 0 is shorthand for primary. So we're just saying this is the primary structure, which is that you have these particular amino acids in this particular order going across there. And remember, for each letter, this is just shorthand for an entire amino acid, an entire little chemical structure. And these little chemical structures are going to be lined up in this particular way to spell out a protein. Now we can take these letters, and we can actually compare the letters of our protein to the letters of other proteins in the world. And proteins with similar letters are probably going to do a similar job, and they're probably going to form a similar shape. That's the neat thing about biology that people have realized fairly recently. And so, yeah, from this particular structure, you could look at it, and this is just a tiny little part of the structure of this great big protein at the bottom. But you could look at it and say, oh, this is similar to some other proteins that do a particular job that's called a nuclease, which is they take nucleic acid, RNA or DNA, and they cut it. You're like, okay, that's fine. That gives you some information about your protein. Second level. All right. So we've got right here a little piece of the protein, and let's see, we're coming out of domain 2 and into domain 3, something like that. But anyway, this is showing that the protein is a mix of secondary structures. And so this chunk here will be an alpha helix, great. And let's say this chunk here and this chunk here will both be beta strands, great. And so we'll label that alpha 1, beta 2. And these are just that same alpha helix. It means the protein backbone is going to coil up. And if you want, you can look at a little coil down here. That's what an alpha helix, that's one way that we can visualize an alpha helix, one way we can make it look. It actually just looks like a collection of atoms roughly arranged in this order, but I think visually it stands out a little more this way. And then beta strands, like these, beta 1 and beta 2, are going to be, let's see, these things that are shown with arrows. So here's 2 little beta strands, just like the ones we're talking about. Or here's 3 little beta strands, so these are making a sheet. These are making a little larger sheet. Here's another sheet. Here's a long sheet right here. But yeah, now you can see them. All right. Third level of structure is the three-dimensional shape. So these are all the coils, the alpha helix, and all the strands, the beta strand here, and everything in between is this random coil stuff. And so you got these little turns and loops and coils, and everything that's not diagrammed out as beta strand or alpha helix is going to be assumed to be random coil. And so from the three-dimensional shape, you can say, well, you know, it kind of looked like a nuclease up here that was a sort of a mix of alpha helix and beta strand and random coil, about an even number of each. And then at this level, you can say, oh, wow, the whole shape of it, when it folds up, it looks like a particular kind of nuclease that cuts in the middle of the nucleic acids. We call it an endonuclease. And if it cut at the end, we would call it an exonuclease. And it looks like one that somebody found from a frog called Xenopus, which is a famous laboratory frog that everybody likes to study. And the overall enzyme is called X-endo-U. That's its name. Okay, and then fourth level structure. All right, so we've got those three-dimensional structures, and now we're showing it in a different way. We're not showing the individual beta strands and alpha helices anymore. We're just showing what the surface would look like with all the side chains on and everything. We were just looking at the backbone before. And so this is the surface, and here we're just showing that there are actually, these things come together and they make a machine, just like we were talking about. And it actually takes 6 of these proteins to make the machine. So the purple one, the light green one, the light blue one, the medium green one, the gray one, and the orange one. And they've just highlighted some parts of these in red here, but forget about those. These are the 6 copies. Each one of these is the same protein. It's another copy of this protein, and they're all folded together. And so this is the quaternary structure, which is based on stacked up versions of the tertiary structure, the three-dimensional structure, which is based on those little beta strands and alpha helices of the secondary structure, which are formed by the primary structure, which is the amino acids in the order in which they appear. So this entire great big molecule, you can call it a macromolecule, is made of 6 protein chains and all of their second and third order structures all folded together. And so that's what a protein actually kind of looks like if you're able to walk up to one. If you could make yourself really tiny, this is kind of what you would see. Okay, so these are those amino acids again, and the amino acids have different groups on the sides, and the different groups on the sides give each amino acid a different ability to form a beta strand or an alpha helix, or to do certain kinds of chemistry if we're talking about an enzyme. Now let's look at what this actually means. So we're going to start out with a particular little protein. And so in single letter amino acid code, it's going to be Q, A, P, A, Y, K, K, etc., just like that. So what is that? 5, 10, 15 amino acids. Nice short little protein. Easy enough to conceptualize. So for each of those letters, each letter actually stands for an amino acid. And so I put the amino acid underneath each of the letters. So Q is glutamine, and you can remember it as the Q-tamine. Yeah, that's how you remember that it's Q. It's dumb, yeah. But whatever, you have to do dumb things sometimes. And so A is alanine, P is proline, Y is tyrosine, K is lysine, because there's already a leucine that got to be L. Here's leucine that got to be L. E is glutamic acid, because the acids are D and E for whatever reason. And S is serine. And so you join these things together by joining up the carboxyl end here with the amino ends down here. And you're going to lose that OH and this H, and boom, those are going to click together, click, 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 to form something that basically looks like a charm bracelet. It's going to have a backbone, and it's going to have different, what we call side chains, which is going to be the group to the left side of each of these molecules, and that'll be sticking out, and that'll give it its properties. Okay, so this is looking at the primary structure. It's these particular amino acids in this particular order. What does that mean to you and me? Not a whole lot, but if we're building proteins, if we're working with proteins, we got to understand it at this level. Okay, now let's take our protein, this whatever, QAPAY, KKAK, KKLAES, KPA, whatever, KKLess. This is how it folds up in 3 dimensions. And so over here you can see I've color-coded the amino acids and put them in their order. So Q is Q, A, P, A, Y, K, K, A, A, K, K, L, A, E, S. And you see they're coiled up. It's maybe a little hard to see here, but they're actually coiled up into a little coil. And so here we're just looking at the backbone. Over here we have the backbone plus the hydrogens are shown as those little tiny balls at the end of the sticks. It's a little simpler, I think. And you can see on the backbone there's this repeating pattern where you have the nitrogen, which is the amine, joined onto this double bonded oxygen on a carbon, which is the carboxyl. And that's that linkage that I told you. And that's between every single amino acid. They're linked up with a carboxyl joined onto an amino group. That's how protein works. And so there are different ways we can look at a protein, and eventually these are all they'll show you different aspects of it, but they all kind of mean the same thing. So if we write it like that, we're just saying this is the primary structure of it, and you're going to have to imagine what it looks like in 2 and 3 dimensions. Or we can show it like this, which is kind of like a 3-dimensional view, but we've blown it apart and shown the primary structure on there as well. Or we can show it this. These are the key atoms of the backbone. Or we can just say, well, it's an alpha helix, and we're going to draw an alpha helix as a twisty, coiled up arrow. So you can just look at a protein and see, oh, this chunk is going to be alpha helix. And in this one, I coded it from red at the start to blue at the end of the protein. Or we could look at that same alpha helix, but we could look at the overall shape, or like what would the outside of this look like, without knowing what's going on the inside of it. And so it's going to look kind of like this. It's like a sort of a bumpy looking sausage. I don't know. Like a chocolate turtle or a bit of peanut brittle or something. This is what proteins look like. They're bumpy things. And this is, again, the backbone from inside of there, but it's just showing the same thing that we saw before. Just I've color-coded it by what order we are. So this is the start, this is the end of our protein. That's what proteins look like. That is how the structure looks. This is, this is what we're going to be dealing with. We're going to be making proteins. We're going to have proteins binding to other proteins. We're going to have molecules binding to little pockets in proteins. And yeah, I just wanted you to have a little basic understanding before we went into stuff like that. Thank you very much.